Assalamu alaikum students, I am Gudala Yasmeen and I am teaching you the course of BA Zoology semester 2. Title of course is Principles of Animals Life. Topic of my presentation is Inheritance Patterns. Let's start today's lecture. Patterns of Inheritance. Genetics is the study of transmission of biological information from one generation to the another generation and its expression in an organism. Modern genetics begins with the work of Gregor Mendel. Gregor Mendel was born in 1822 in Europe. He was the son of a farmer and grew up learning about the cultivation and propagation of crop plants. He laid the foundation of classical genetics by formulating two laws of heredity. In 1857, Mendel began experiments with garden pea, Pisum sativum. He published his work in 1866, but his contemporaries did not recognize the importance of his conclusions. While in 1900, the rediscovery of his work propelled genetics into 20th century. Study of the pattern of inheritance. Gregor Mendel selected pea plant for his experiments because of certain reasons. Pea plants grow very easily. It possesses distinguishable characteristics like the seed shape is either round or wrinkled, the color of the seed is either yellow or it's green, and the height of plant is tall or short. It can also be easy to control their mating. Before studying the laws of inheritance, we must be familiar with the terminology used frequently during this study. Now, what is allele? Allele is the alternate form of a gene. Dominant alleles, these are the alleles that code the trait that exert its effect whenever present, and they are symbolized by capital letter, for example, capital B. Recessive allele, they code the trait that is masked by if a dominant trait is present, and it is symbolized by a low-case letter like small b. Genotype, it is the genetic makeup of an organism. Genotype is homozygous when two alleles that are same, for example, either they are capital double B or small double b. And they are heterozygous when the two alleles are different, like capital B and small b. Phenotype, it is the observable characteristics, like the brown eyes or brown hairs, tall or a short person. Generation, it's a group of organisms which are born around the same time. P generation stands for the parental generation, while F1 is the offspring of the parents, also known as first filial generation, and F2 is the first generation of the offsprings, also called second filial generation. This table also shows definition of some other terms like chromosomes. These are the macromolecules found in the cell consisting of DNA, protein and RNA. Gene, it is the unit of heredity of a living organism. Locus, it is a specific location of a gene or a DNA sequence or position on a chromosome. Alleles, as previously mentioned, is one of the number of alternative form of the same gene or genetic locus. These terms have been mentioned in the previous slide. Now, true breeding fly. What is the true breeding? It is a pure breed. It's an organism that always passes down a certain phenotype trait to its offspring. Wild type are the most common phenotype in nature, while mutant, it's the physical or the genetic difference due to mutation in a gene. Now, Mendel inheritance pattern. The fruit fly Drosophila melanogaster is selected to study these inheritance pattern because of its short life cycle, easy to handle, and possessing distinguishable characteristics. The figures show the normal fruit fly called the wild type with red eyes. Male fly can be distinguished from the female fly by having a round abdomen tip with wide dark bands and having 6 combs at their forelegs, while the female flies have pointed abdomen tip with light narrow bands. The chromosomes of the male and the female also show differences. Drosophila has 4 pairs of homologous chromosomes, 3 of which are similar in both sexes, but the fourth pair is very different. In female, this fourth pair is having 2 similar rod-shaped X chromosomes, while in male, one of the chromosome is rod-shaped X chromosome, but the other one is J-shaped called the Y chromosome. Law of segregation. It states pairs of hereditary factors are distributed between gametes during gamete formation. According to this law, 2 coexisting alleles for each trait in an individual separate from each other at meiosis, so that each gamete receives only one of the 2 alleles. These alleles unite again at the random fertilization of gamete when zygote is formed. A cross was made between a wild type and a mutant fly to illustrate the Mendel principle of inheritance. This is a cross involving a single trait. In parental generation, wild type fruit flies were crossed with the flies having vestigial wings. The offspring of this progeny, the offspring of this cross have all wild type wings. If these flies of F1 generation were allowed to mate among each other, then it was observed that approximately 1/4 of the flies were having vestigial wing and 3/4 of the flies were having wild type wings. So the ratio of wild type flies to vestigial wing flies in F2 generation is approximately 3:1. Alleles for the wild type wing is dominant over the alleles for vestigial wing. So in this previous example, the flies of F1 generation have the same phenotype as one of the parent, means all of them are wild type, but they are different genotypically because they carry both dominant and recessive alleles. They are hybrids. And in this cross, since we are dealing with only one pair of a gene in a single trait, so it is called monohybrid cross. Original fruit fly cross. In parental generation, wild type and vestigial wing flies are homozygous as they carry 2 identical alleles for a given trait. These are obtained only when true breeding flies are crossed. While all the F1 flies of F1 generation are heterozygous, mating of the F1 flies with each other result in the ratio of 3 wild type flies to 1 vestigial fly in F2 generation. So this 3:1 ratio result when 2 alleles, each of which is present in the separate gamete, combined randomly at fertilization. Punnett square. Punnett square is used to predict the result of the cross of 2 F1 flies. The kind of gametes that each member of a cross produce are determined and placed along the axis of the square. These are the male gametes, and on the upper half these are the female gametes, while the combining gametes in the interior of the square show the result of mating. A phenotypic ratio of 3 flies with wild type wings to 1 fly with vestigial wing is clear, while the genotypic ratio represents the result of a cross according to the relative number of the progeny in each genotypic category. That is, 1 homozygous wild type is to 2 heterozygous wild type is to 1 vestigial wing. Law of independent assortment. It states when 2 contrasting pair of traits are followed in the same cross, their alleles assort independently into gametes. In case of Drosophila, crosses are made using the flies with 2 traits. That is the flies with vestigial wing and the sepia eyes, and the other group is the flies that are vestigial, that are wild for these characteristics. There is sepia eyes. What are sepia eyes? These are the dark brown color, and wild eyes are of red color. Punnett square for a cross involving 2 traits. This shows every gamete has only 1 allele for each trait. In this cross, flies in the parental generation are homozygous for their trait. Each parent produces only 1 kind of gametes having 1 allele of each trait. As a result of fertilization of their gametes, all offsprings are heterozygous for the both traits. All the F1 flies are phenotypically wild type it means wild type eye are dominant over the sepia eyes. F1 flies are hybrids, and because the cross involved two pairs of genes and two traits, so it is called dihybrid cross. In this cross, phenotypic ratio is observed as 9 wild type flies with wild type eyes, 3 wild type flies with the sepia eyes, 3 vestigial wing flies is to wild type eyes, while 1 vestigial wing and sepia eyes. Conclusion. The distribution of genes of one trait did not influence the distribution of gene determining the other trait. It means gamete with wild type wings for the wing may also have either the wild type or sepia gene for the eye color. Gametes of an F1 fly all show the combination of eye colors and the wing condition, and all the combinations are equally likely. This clearly explains Mendel's principle of independent assortment, which state that during gamete formation, pairs of factors or genes segregate independently of one another. Testing phenotype. Now we have to test the genotype of an individual which show a dominant phenotype. A cross is made, which is called test cross. It is the mating of an individual showing dominant phenotype with an individual showing recessive phenotype. This cross find out the homozygous or the heterozygous nature of genotype. Test cross. This is a test cross for dominant phenotype wild type wings. In case number 1, if a test cross with a fly homozygous for a dominant trait was made, it always yield phenotypically dominant offsprings. While in the second case, a test cross with a fly heterozygous for a dominant trait always yield 1:1 ratio of phenotypically dominant, that is wild type wings, to a phenotypically recessive offsprings, that is vestigial wings. Students, this is the end of today's lecture. Hopefully you fully understand it. Thank you. Allah Hafiz.